Okay, so we are in the home stretch here, and now it's time to discuss how to finish our cleanup. And the last thing we need to do here is take care of these gaps, right? So we have done helicase, topoisomerase, SSBPs, primase, DNA polymerase 3, DNA polymerase 1. 1, 2, 3, 4, 5, 6 different things, 5 enzymes, and SSBPs, which are in eukaryotes only. One enzyme left to discuss, and it's the enzyme that takes care of the gaps. And that enzyme is called ligase. Ligase seals the gaps. But ligase has an interesting restriction. And that interesting restriction is that ligase can only seal the gaps on either side of a DNA fragment left from polymerase 1. In other words, if we look real quick, at a little section of our strand where we have our bits of DNA. Put that too close together. So this is our section of the lagging strand, because there's two bits of DNA where we had to remove primers. And then we've got our section of our leading strand up here with our DNA from DNA polymerase 1 and all that DNA from polymerase 3. And we have to seal a gap. And I like to conceptually just draw ligase in a way that explains its restriction. So here is ligase, right? And it can only seal gaps when there's, space, DNA on both sides of a gap. So can only seal gaps when you find DNA or on both sides. So we'll just draw it right here. Oh, look. Right? We've got DNA on this side, and we've got DNA on this side. So check, check, and we can seal that gap. We'll just gap sealed. Uh-oh, what about over here? All right. DNA. Check. But oh no. No DNA. So ligase can't seal that. So because it can only seal gaps when there's DNA on both sides, ligase cannot seal gaps at the terminal ends. any primers within the strand, especially all those Okazaki fragments. It can seal those. But terminal ends, it cannot seal those gaps. Now, luckily for us, there's going to be an extremely lazy solution. So, let's say this is our full-length DNA strand. Right? So, we've managed to make our full-length strands. But, unfortunately, we can't seal these gaps at the very end. Now, there is, in fact, a very lazy solution. Because remember, the universe trends towards maximum laziness. And the laziest solution here is literally... undo is literally to just cut those bits off. Bloop. Bloop. This back. Right. And just snip that right off. Can't seal the gap? Well, cut it out. What this means is that with every replication, don't know what I was spelling there. With every replication, chromosomes shorten by 5 to 10 base pairs. Doesn't sound like a ton, but replication happens extremely often throughout a cell's life cycle, and humans especially live a pretty long time. So it is entirely possible that cutting these tips off, right? So we cut the ends off where ligase couldn't seal gaps. And so that can get very bad. You can start to lose actual coding genes. So the simple solution on how to protect your DNA when every replication causes it to shorten by 5 to 10 base pairs, right, is to have all these important genes. So, you know, you've got gene 1, you know, gene 2, right? All these important genes in here. You don't want to be losing these genes. So all you got to do is have a bunch of nonsense DNA at the ends. So non-coding nonsense DNA at the ends, and that protects the genes from getting cut. So these non-coding nonsense DNA bits at the ends are called telomeres. And the telomeres get cut away with every rep in order to protect the genes. So, and that's the end of DNA replication. Hooray! Pretty cool. It's thought that as you age, there comes a point where you run out of telomeres, and it starts cutting genes that you actually need. And part of the breakdown of the body with aging is due to the breakdown of genes once replication starts actually cutting out functional genes. There's correlations between telomeres at a certain length threshold and longer lifespans, but it doesn't directly correlate like telomere length to lifespan. Like a short telomere length doesn't guarantee a short lifespan. A super long telomere length doesn't guarantee a super long lifespan. But there's like this little threshold where if your telomeres are this long, you're probably going to be an animal that lives a relatively long lifespan, which is pretty cool. Not tested over. Reptiles, which tend to live extremely long lifespans, especially relative to their size, tend to have, especially tortoises, have an enzyme called telomerase, which rebuilds telomeres, which is pretty cool. So it's thought that if they didn't have to worry about injury, disease, competition, starvation, those sorts of things, tortoises could live even longer than the like Galapagos tortoises and Aldabra tortoises and those things. They've been found like 200 years old, 300 years old. It's thought that they could live much longer if we could solve other problems for them. So, and also, even as they age, their brains don't turn to mush, and they don't experience a lot of the same senescence issues that mammals do. Pretty cool. None of that telomeres affecting health stuff and length of telomeres and cool reptile telomerase stuff is going to be on an exam, but fun facts are fun facts. All right. Replication done. Woo-hoo-hoo.